Okay,
Looks good.
okay, sounds great. Welcome everyone to day three, the last day. Today will be a shorter one. For the last 2 days, we read papers, we did coding sessions. Just as a review of what we did yesterday, let me push this around. Um, we did PCA, we plotted PCA, we did UMAP, and neither of those are magic methods, although they're quite complicated. Um, they're not magic in the sense that they cannot separate noise from signal. They are there to find patterns, but patterns could be signal or could be biological noise. And so oftentimes the input to these methods are also important, although people gloss over it from time to time. So anyway, we need to normalize our data depending on what we think is the right way to do it. To remove as much as possible technical artifacts. And there are typical normalization steps people use to do spatial transcriptomics for single cell transcriptomics. Um, so usually we need to normalize raw counts by cell size, meaning you don't the absolute number of how many genes in total being captured in a cell doesn't make much sense. Um, why is that? Because a cell could be small. Sorry, cell sizes could be different, but that cell sizes is not captured by either sequencing or imaging technology. By sequencing, there is a step of amplification. You amplify all the transcripts in the cell. And for imaging, it relies usually on thin sections of tissues, and a thin section contains about the length of a cell. So sometimes you cut through half of a cell, you cut through 80% of a cell. That automatically makes you vulnerable to the cell sizes. Okay, so so if you do normalization, uh, you will find usually it makes a big difference. Yesterday we made this UMAP. I guess many of you have got to this point. Otherwise, you've seen this slide. The UMAP looks something like this. But if you do normalization, so we got that point because we went straight from raw counts, the raw data, how many copies of each gene for any cells you get. Those raw data directly do UMAP, PC and UMAP, then you got this. But if you do normalization just by cell size, you got something different. Okay, so this is something you can try. And if you compare between the two, you see normalization make a big difference. It removes a lot of variations um, within a particular branch. For example, this gene is expressed in all over the place here, but on this tip, it is expressed much higher than here. Is it because a cell size effect? Is it because of a real biological effect? Is harder to know. But after normalization, you see this no longer exists as much as previously, suggesting after normalization, you remove this variation. And at the same time, many other structures reveal themselves more fine-grained structures remove themselves, indicating you likely identify more cell population, more cell type differences once you remove those technical variations. Okay, so this code, you just take one line to do the normalization. I provided that to you already for yesterday's. So after UMAP, there's a normalization step. You can then reapply UMAP. Okay. So today let's move on um, to the last bit of analysis we're going to do together. We talked about how to plot a gene, how to do dimensionality reduction, and are there different cell types? can we put a label on cells? We already see from the UMAP space that there are clusters of cells, each separate from each other, suggesting they are different types. Can we apply a clustering algorithm to call them out? And after clustering, after getting the cell type label, we can do pseudobulk to identify cluster centroid for each cluster. So that's another thing we're going to do today. And as a bonus point, you can feel free to explore once we get the cell types. Can we ask which pairs of cell types interact more in space than others? Okay, so for coding, there's just two exercises today. First, we'll do clustering. We'll do the simplest possible clustering called k-means clustering a little bit. And then after clustering, each cell has a cluster label. We will try to calculate the cluster centroid gene expression, the mean gene expression for that cluster on average for all the clusters. That's called pseudobulk gene expression profile. And that'll be all for the day. And I'll leave out some empty space in the end in case you wanted to try the last exercise or you have your own questions. Okay, so should we, yeah, go ahead. Yes, yeah, I can show you. Yeah, I can show you. Definitely. And it's going to be provided in your homework solutions as well. So the code is easy. It takes all of these, the key code is just a few lines. 1 or 3 lines of code can do all of these, but you just need to know what to write, I guess. Okay, k-means clustering. Mm, so we will do this together, but let's talk about the concept for now. We had this data. We plotted them by gene expression yesterday. We showed that you can do UMAP based on the similarity of gene expression, but we can also label cells by discrete labels. Here, each cell is colored by one of the 20 colors shown here. So each of the color represents one cluster we identify from the data. And here I plotted their spatial distribution. Here's their cluster distribution within the UMAP space. Okay. So k-means, it looks like they make sense. For example, a group of cells in the UMAP space that are similar to each other share the same color, indicating the k-means algorithm identified them as the same cell type, which makes sense, right? So what does k-mean do? What does it, as a clustering method, you try to figure out this. You try to group similar cells together by their similarity in gene expression. Okay, so in order to do that, you need to first specify how many cluster you want to get from your data set. Initially, your data you just have 80,000 cells, but you now need to specify, do you want to get 20 cluster from the 80,000 cell or just 3 cluster from 80,000? This K means the number of cell types you want to identify. Of course, it's your choice. The algorithm doesn't figure it out for you. So this is a character for any clustering algorithm. There's always some implicit or explicit variables that let you specify how many cell types you want to get from the data. Okay, that's a choice you have to make. So once you make that choice, say you wanted to identify 20 cell types from it, it automatically identifies what are the best 20 cell types possible. By best, they mean that you wanted to find 20 groups such that within the group, cells are pretty similar, but between different groups, cells are quite different. So you want to maximize the difference between groups while minimize the difference within group. And that's an optimization. Mathematically, you can define and create and have an algorithm that does that. So the algorithm is also a typical trick many algorithms will use is called iterative algorithm. So the way they do it is to try to solve a chicken and egg problem. You first randomly assign cluster and cluster centroid. Okay, so you don't know which cell belong to which cluster. You don't know what the cluster centroid is. So you first randomly assign everything. So once you have a cluster centroid to begin with, then you assign cells to the closest possible centroid you randomly initiated. So now you have an updated version of cell type label that allows you to recompute a cluster centroid that's closer to the truth. And then once you recomputed the cluster centroid, you can update the cluster assignment for each cell to reassign it to the closest centroid. Okay, so what if you iterate repeatedly enough times, there's guarantee that you will converge to an optimal solution. That's 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 mathematically proved and what tight proved, and there's no worry that it won't reach that. Okay, so so that's it. So let me make do an example for that. So I'll show you an example, a coding example. Yeah, go ahead.
Is there any way to estimate AK? Because sometimes you don't know
Yes.
the exact number. You have to see.
Yeah, there is way, but there is no ground truth way, if I put it. So here everything is tight, and you can objectively decide once you decide a k, but the choice of k is never objective. If that makes sense. It depends on your choice, your circumstances, your you can design a metric yourself in order to convince, or you can try a different k, see which k performs the best by your standard. But there's no in general no objective way of doing clustering. That's one difference from so these kind of learning. So in machine learning, right? There's three main categories of machine learning. Supervised learning, you must supervised learning means if you have data x and y, you wanted to learn a function such that you can predict y from x, right? You go from x to y, try to learn a function f. That's called supervised learning. That one is objective in the sense that you can always measure your error, right? And then did you learn well in predicting y? You just test it. That's supervised. This kind of things are called unsupervised learning, meaning there's no ground truth. Are there ground truths 20 types to begin with? There's no. I guess in some situation you can simulate and see. So, but if you know there's only 3 cell type to begin with, maybe in certain circumstances you can try to design, but there's no classic way of so there's a lot of research trying to argue, I guess, but for single cell, I guess in this biological context, people have been arguing about how many cell types are there in the tissue in the brain for a long time. It depends on what you mean by the cell type you're trying to define. So actually, yeah, some people think there's only 3 types, some others found a reason from 5,000 from the whole mouse brain, but even that is debatable. And so, yeah. And at the end, you could have argued that every cell is a bit different from others if you can robustly identify the difference between a single cell. So you can say all 80,000. But so actually, it's a good point. K, what's the range of K? K could be as small as 1, as large as the number of cells in your data set. Okay, anything in between, the algorithm can generate something for you. Do you believe it? You need to think about it. Okay, but let me show you an example. These are good questions. First on the board, say I generated 3 clusters of data. So I don't know, I measure, say, measure the length of something on length of 3 different objects. Each has a fixed length, but maybe they expand or contract a little bit from day to day because of temperature differences, right? And then I measure it on the 3 different objects on 3 different days. That gave me 9 in total, 9 different readings, right? So, and all the data, I just plot all the data on the sheet, and we know the ground truth is 3. Okay, 3 objects, but there's an object whose length hovers around 10 millimeter, whatever. But on a different day, sometimes I get 11, sometimes I get 9. So that's 3 readings I got from that object. Okay, another object, maybe I shouldn't say length, but for the simplicity of calculation, it hovers around 0. Sometimes I get 1, sometimes I get -1. That's the second object. Okay, so there's a third one. On average, you get -10, but sometimes you get -9 and -11. Okay, so I have 9 data points. That's my here. Let's assume that I wanted to apply k-means algorithm to group these to group these 9 data points into three clusters. K equals 3. Apply k-means, so k equals 3. Will it work? Will this procedure of randomly starting a centroid and then iteration, will it work to split them into 3 groups? That's the question. Okay, so we'll just do a k-means together on the board, because in this case it's relatively easy. So the first step. What's the first step? Compute cluster centroid, but first randomly start with a guess of cluster centroid. Because k equals 3, we need to come up with 3 centroid location, 1 for each group. So let's just start randomly. Okay. Could you let me know what's the month of your birthday? October. Okay. So one of the centroid will be 10. Okay. How about you? August. Okay, 8. Another central will be over here at 8. Another one, maybe July. Oh, 7. So let's start from -7, because there's no negative. I guess you can start up there, but it take more iterations. But just to see. Okay, we start with 3 centroid location. Next, so once we get the centroid, now what's the next step? We can assign individual cells to the centroid. Okay, to the closest centroid. Okay, so here, obviously, so maybe I should use 3 different colors for maybe this is a square. This is okay, square, triangle, and rounded. Okay, so in the first round, this 11 data point will be assigned to the square, right? And this 10 data point will be assigned to the square. This 9 is equal distance between these two. But for the sake of I don't know, just randomness, let's assign it to the 8. It could be assigned to either one of them. Okay, sorry. So, that's the first one. Okay, so then this 1 will be assigned to, so 1 8 minus 1, 7. Yeah, closest to this triangle. 0 will be closest to -7, I guess. Yeah, and then -1 will be closest to -7. So all of these below will be closest to -7. So this is -7. So all of these. Okay, so because of the initial random association, we ended up with these 2 points belonging to this, and these 2 belonging to these, and all of the below belongs to -7. Okay. Okay. So now starts the next iteration. What's the next iteration? We want to recompute the cluster centroid given the assignment, right? So now these 2 belongs to the square, which is 10 and 11. So their new centroid location will be 10.5, right? And these 2 belongs to the triangle, which is 1 and 9. So their centroid location would be 5, right? 1 plus 9 over 2. And these 5 belong to this one. So so -30, -31 divided by 5, roughly -6. Okay, so let's say updated one is -6. So well, this closer to okay. So now the centroid location moved. 10 moved to 10.5, 8 moved to 5, -7 to -6. Is that correct? Yeah, that seems correct. Okay, now comes the next iteration, reassigning them to centroid, right? Looks like we now look at these 3 time points because they are all closest to the centroid than this because it's no longer at 8, it's at 5. 9 minus 5 equals 4, which is much further away than this. So all of the above will now be assigned to square. Okay, and then all of these guys, 1 will be 1 is closer to 5, so we'll be assigned this. 0 is closer to 5 than -6. -1 is closer to -6 than 5. So -1 will be still assigned to this centroid below. Okay, then all of these will be assigned to -6 centroid. Okay, this is iteration 1. This is iteration 2. You see, after 2 iterations, things has already improved, right? It started randomly, and then even 2 centroid are so close together by chance. Now things are sorting out, and then now we proceed with the next iteration. So the square one, these 3 together, the centroid will be 10. Okay, and for these 2, the centroid would be here 0.5. For these 4, the centroid would be -31 over 4. What's 31 over 4? 8, yeah, 8 something, -8. Okay. Now we do it again. We just need to be patient. The computers can be very patient. So we reassign them. So these 3 will stay the same. And now the centroid is at 0.5. So all of these is closest to 0.5. And this -8, all of these is closest to -8. Okay. Okay. Now what do we do? we could stop. We know we are already got to the real solution, right? We got to the, but the computer doesn't know it's the correct answer yet. It needs to iterate it again. So let's do it again. Next iteration, we will recompute the centroid here. These three belong to the square, so the square centroid will be 10. These three belong to the triangle, so the triangle will be triangle centroid will be 0, and these three belong to the the centroid will be -10. Okay, what do we do now? We reassign. So after we reassign square, 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 we get a triangle, 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 and circle, circle, circle. Okay, now the algorithm realized that the answer you achieved for assigning cell for this iteration is the same as the last iteration. Now it stops. You know, you stabilize. Everything will, if you keep iterating, everything will stay the same. The algorithm reached convergence, what we call it. And this convergence automatically also ensures that you reach the best solution given the data. Okay, so that's k-means algorithm in a very simple case, but it also applies to very complex case, and they can operate very efficiently in computer programs. So you can tell them they're much more efficient than me doing it on the board. But here we do it on the board just to show you how simple this procedure is. Each step is a simple procedure. Anyone can do it, but you just need to do many times, do the same thing over and over many times. Okay, so we already did that demo on the board, and let me show you how to do it on in the coding environment and on the simulated data, 3 groups of cells and so. Okay, so let's move together to the shared collaboratory workshop repository. Again, the same link as yesterday, and then go to exercise 4 and 5. Okay. Okay, so so because it takes some time to set up, so let's set up together. I'll give you 5 minutes, and while I also do it, just again, as a review, we need to install some package, and that takes a few minutes, and then some functions, and load the data we were working with yesterday, and check they are all right. All of these, okay. So you can run all the way until exercise 4, clustering. So let's just set that up. Over. Okay, so my code is still running. That's because first it takes about a minute to install this package, and then it takes some time to compute UMAP. Just as a review of yesterday, this is all we learned yesterday. I just provided the solution to you. Here I'm still running for the UMAP. It takes about 3 minutes. Hopefully that's my note. It takes about 2.5 minutes when I run it half a year ago. So and then after that finishes, we should be able to get to the same page here. Oh yes.
Normalizations.
The normalizations we did here. Yeah, let me explain that, right? Or is that, oh yeah, a question? Okay, I see. Okay, where did we do normalization? Oh, now my yes. Okay. So here we get the data. The data is a count matrix, right? That's called features. Gene by cell. Here is a cell by gene count matrix, right? And I did normalization by size. And then what did I do here? First of all, features sum over x equals 1. So just to explain, here you have a big data matrix. The columns are, so the rows are different cells, cell 1, cell 2, and so on. Columns are different genes 1, 2, 3, okay? I first sum over all the genes for each cell. Okay, so that is my COV, coverage, COV, which is just sum over all the columns row by row. What does it mean? It calculates all the transcripts that's captured in any cell, right? And then what does this do? It takes the median of the counts, median of the coverages, median from the coverage, because you can imagine some cells have more counts, some cells have less counts. The median is on average, a typical cell gets how many copies of transcripts, okay? So then I just divided the original counts by a factor of this factor is coverage over median coverage. Does that make sense? So if a cell is above the median, you try to tune down all of their values. If a cell is below the median, you tune up their values so that all the cells have equal number of transcripts after normalization. They all have the same number that is set by the median. Okay? So that's normalization. And then after that, I did log transformation. Remember, yesterday when we did the plot, the distribution is very screwed, like this, for a gene called SCLC70A7, with many cells having 0 counts. A few cells have like between 0 to 100 counts. Very few cells, but have very high counts, up to 500, right? If you directly use this feature to do all these algorithm, PCA and UMAP, if all the genes are like this, usually it's the algorithm doesn't like that. All of these are a few outliers can influence a lot on the mean value. They don't like it. So a typical way people normalize things to make this distribution more well-behaved is removing the influence of outlier is by doing a log, right? Log 10 means log 10 plus 1, actually. So this 0 will become 0 again. This 100 or roughly becomes 2 because 10 to the power of 2 is 100, okay? And then 1000 will become 3. So 500 is not much. 500 is about 2.5 or something. It removes the influence of outliers by a lot. This is something you can do, or this is quite typical, I would say, when you apply all these algorithms. But there are also, you can also do without it to some degree if you remove outliers, or so anyway, so then after that, after normalization, we did PCA, we did UMAP, and then we can plot the UMAP and plot the UMAP side by side with spatial distribution, cells colored by the expression of one gene, and then now we're here for cluster, right? Okay, so I'll let you apply k-means on the real data, but let me show you applying k-means on some random data. Okay, so let's ask ChatGPT. I wanted to generate first three clusters of cells. So make a scatter plot with 3 clusters of points. See. Okay, I'll trust it and copy the code over. Invalid syntax. Did I copy everything? You see, they created 3 clusters of points here for me. But at first, let's see. So they already, so let's look into their code. Okay, they generated cluster 1, 2, 3, each having, I believe, 50 points, and then they stack them together into this data matrix. So we know that the first 50 data points belong to blue, and then the next 50 belong to green, the last 50 belong to red. Okay, so but let's assume we got this data with no labels. So what I wanted to show is that initially, before applying k-means, we got some data that looked like this. Okay, initially we just have three groups of cells. We don't know there are 3 groups of cells. We just got some cells, but by eye, intuitively, we think there should be 3 clusters. So let's apply k-means with k equals 3 and see if k-means identify those clusters. Okay, so then let's ask apply k-means clustering to assign each data point a cluster label. Okay. Okay. Okay, it did that. First of all, let's check what they did. Okay, so now they applied k-means clustering on 3 groups of data, and they also showed you where the centroid is. So they show you 2 things, the cluster assignment and the centroid for each cluster after applying for k-means. Okay, now it becomes effortless to let AI told you what to do, but do we understand what's going on behind it, right? So here, first of all, let's just look into their code. This is some random seed to nail down reproducibility because there's some randomness built into the k-means algorithm. Let's fix that. And then generate 3 clusters. We talked about that. And combine all of these clusters together into this data variable. Here they applied k-means. Okay, so you specify an algorithm k-means with 3 cluster and the fixed random state, and then you send your data into the k-means to let it predict the results. The results are in this thing called labels. Okay, so let's do it. Let's check what this label variable looks like. First of all, first of all, let's print what the shape of our data point is and what are the labels. Okay, so it's 150 by 2 is the size of our original data because it has 150 data points. 2 is two features because we are working on a 2-dimensional data space. And then the labels are also 150 because each cell or each data point gets assigned a label, right? Then we could look into what the labels are. So you see a lot of different 2s in the beginning, a lot of 1s in next, and a lot of 0 and 1. It just means, so there are 3 unique numbers, 3 unique labels, each belong to one of the cluster. They happen to think the first 50 cells or so belong to the same cluster, which they assign a number called 2, then the next 50, 0, then the next 51. Okay, so that's k-means algorithm. So that's the labels. So then if you make a plot, AI already made a plot for us where you first plot the data and then you plot the centroid and the data is colored by labels. That's what you see, right? So these data are colored by their assigned labels and all make sense. Okay? That's how you do k-means. So what do you need to do, right? You don't need to generate random data and compile them together. You already have the data, right? Then you just need to know how to call k-means and specify the number of clusters here, then send your data in to produce a label. And once you have a label, you can plot them in your data set, okay? So that's the first coding exercise. I'll give you 10 minutes, and if that's not enough, we can have we can give extra time. Okay. So remember we have our shared Google Slides. You can copy this over. Let me copy my over. This is the ring. Okay, this is my k-means results. Okay. Yes.
How you decide k-means versus
Yeah.
and then you like decide.
Yeah, you decide.
You try both and you see.
Yeah, it's usually for most purposes either way would.
Yeah.
Yeah. Each emphasizes a different thing. This is simple and straightforward, quick. Hierarchy takes a little more time, but it gives you a trees, and that's the question.
Yeah, because in the most range practitioner, they use that, but I think maybe you have to in the too many cells. One.
Yes, I believe so. But their hierarchical is more, it's a hierarchical of trees, not necessarily trees. So there's simple ways to do hierarchical. You're given a set of data. You just start from the ground up, compare what are the most similar to where to them. So that I believe is similar to starter. I think Talon is more sophisticated in the sense that I think they first divided cells into a few major groups. That's the first layer of hierarchy. With those, think of it as you have a big data set, choose a small case, although they didn't choose a different algorithm, Biden or something, but still choose a small case. And then for each of those, they redo all the go over all the analysis. Yeah. By redoing, I mean that they don't start from the same matrix. Recluster, actually before recluster, you select highly variable features and all these things are important actually. It's a lot of key applied on different subsets.
Yeah.
Then before king, there are different normalization, different selection. So this takes one line, right? There's over 800. But once you know this, you can get your party. You combine these different tools together. That's where it takes time.
Yeah, and then my discussion. People knows more about and one person decided how this version.
Once these basics are easy, those three discussions in integration. Okay, how are we doing here? Yeah, looks like there's some progress being made. That's great. So if you run all the way to this point, it shows you. So we already provided you with code to do k-means with k equals 3 on this data. And does it make sense that the three clusters make sense? Roughly, they do. If you're a neuroscientist, they correspond to roughly excitatory neuron, inhibitory neuron, and non-neuronal cells. Although, and their distribution in space also makes sense. But what this UMAP indicates is that within each within each group, there seems to be finer structure that's not captured by k equals 3, right? So that's when you can try more k's, and then, for example, this is still sorry, this is still 3 clusters, and but if you try, for example, 20 clusters here, it also makes sense. And spatially, you see these different clusters lining up like a sheet on top of each other, right? On the outer edge of the brain. That's the cortex. And in layers, and that's captured over there. And these structures, hippocampus. And you could try even more than 20, and then that may let you review even finer structures, say, within the hippocampus, and so on. So, yeah, that's something to play with. And good job, everyone. So let's take a 5-minute break, and we'll be back with our last exercise in class. We've done k-means. We will work on top of k-mean, just compute the cluster centroid pseudo-bulk profile for all of these clusters. Okay. 5-minute break. 10:07. Hey. Oh, is it okay? Yeah, no way, no way. Oh, sorry. This is for quick protein study.
Oh, well, I'm in that class too.
Okay.
All that.
Yeah, no problem.
Wasn't. Okay. Thank you.
Okay, so um, let's do our last stretch for the workshop. So we just came up with k-means with k equals 3, and we have 3 centroids. Now we wanted to know what are these 3 centroids, right? What are their profiles? What are their gene expressions? Or if you have 20 clusters, what are their centroid profiles? So this is the centroid profile for our dataset. If you call 20 cell types, 20 clusters, each of those rows is 1 cluster. And we measured 480 genes. So each of those clusters has an expression profile of those 480 genes. So you can summarize their centroid profile in the matrix like this, just as here. Each centroid has a location in this 2-dimensional space. Here it shows each centroid has a location in those 480 dimensional space as represented in this heat map. Okay, just gene expression profile. And then with that's useful because now you can interpret what each cluster means. For example, this cluster 8 seems to be expressing a set of genes highlighted here quite specifically. Then what are those genes? What are these clusters? Where they're distributed? You can compare all these information to learn what's going on in the data, right? So how can we do this once we get the k-means? That's our next exercise and the final one. So for this one, I wanted to challenge you a little bit because so far the concepts are a little hard, but the coding part is quite easy in the sense I provide you with the examples, and you can go all the way towards here. You just need to change the number of k to a different number, right? So this time I didn't provide any code to you. You already have k-means. You know how to do k-means. You generated 20 clusters. Your task is now to get the centroid profile for those 20 clusters with no template. But I did provide you with a step-by-step guide how I would do it. So it may or may not be useful to you, but you could figure it out yourself or whatever ways. So I'll give you 10 minutes to try that and see where you can go from there, and let's see, okay? But that's the last exercise of the class. How can you break apart this task you have once you have a goal? How do you break it apart step by step to get what you need? Yes.
This is
Circular, right? Or what?
no, this bulk, like this pseudo bulk, is what you need to do first in order to be able to compare.
to do to differentially express gene between two clusters.
between all the clusters.
Between all the clusters, yeah, let's say just two
Is this a
clusters.
like something you had to do before or
Not
not necessarily?
necessarily, not
Ah,
necessarily.
okay.
Yeah, usually I would do it, but not necessarily.
So you can go directly to
Go direct-
differentially between them.
Right, depending on what they need, right? So, I mean, but centroid is an easy step. They probably always do it internally or right?
Yeah, that's
So this is cluster 1. This is cluster 2.
Mhm.
Differentially expressed gene means this is one gene, right?
Mhm.
One. And then you have a bunch of cell belong to this bunch of
Yeah.
cell. Yeah. Point bunch of cell and maybe some differentially expressed gene just are they different enough,
Yeah.
right? Yeah. So usually centroid is always a measure, right? So you compute the centroid of this centroid this, and the difference between the centroid is always it's almost always a metric to
Okay.
in either they baked it in or you need to provide it. Either way, it has to it has
But it's kind of
part of the
okay.
So usually it goes beyond the centroid.
Mhm.
They also consider the variation, right? If the centroid is big enough, but they are within each one is
Mhm.
variable, two variable. It's yeah.
Okay.
Yeah.
Thank you.
Yeah. But I guess it depends if you have a few replicates, right? So
Mhm.
cluster 1 have animal 1, animal 2, animal 1, animal
Mhm.
2. That becomes a bit more complex. That's
And it's
so
always like that. We always have
yeah,
replicates.
definitely. So and then to do differential expression, I think you need to do centroid for each replicate. That's what I rather than centroid for the entire thing. It might be tempted to do the centroid of the entire thing. That's okay as a report so afterwards. But during the test, you need centroid for each replicate. Yeah, that's just common sense, right? Because otherwise,
I
so
think adding, because this is not easy, but it's like you can understand when you're working with just one brain data set. But when you start adding replicates and they probably don't behave exactly the same between each other, I think everything gets complex.
yes.
Okay, thank you.
Yeah. Okay, let's let's see. So I saw some progress. This is close enough. But let's do this together. First step. So first step, we need to get this data, right? We have this data cell by gene. Gene is normalized feature matrix. Well, where is that information stored? That's in this variable called features underscore normed. Let's just check that. What this feature underscore normed is. So yeah, it looks like it's our data matrix, right? 84,000 rows, 483 genes, and the genes are not integers because it has been normalized. You could use unnormalized, and the procedure will be the same, but because we know we wanted to normalize our data, and then so we just use that. So that's our data matrix to start with, to work on. Okay, so step one, it says create a new data matrix or a data frame called TNP, temporary matrix, just something we wanted to work on because we want to change the original one with a copy of the original one. The reason we do this is only for is only because we don't want to, if we screw something up, we don't want to corrupt the original data. This is always good to do, right? And it's fine if you directly work on the original data as well. You can, if you made something unrescuable, you can always restart everything all over again. But I don't want to, so I just make a copy of this thing, copy it into this temporary matrix. That's my step one, right? Step two, add a new column in this data. So now we have this data matrix. We wanted to add a new column. Let's add a new column over here. For each cell, this column is the cluster label. We already got the cluster label, one of the 20 for each of the cells, right? We just want this data table to have that information in there. So to add a new column, let's call it cluster, or yeah, cluster. Where was the data label stored? Let's go back and see. In my case, it's this CLSTS cluster. And it only contains 3 clusters in my case because I didn't run the 20 cluster one. So that's fine. So let's add that in. Okay, so now that's step 2. Let's just check if we did that. So if we scroll over all the way to the end, you see there's a new column called cluster. We just added that in. Okay, so that's step 2. Step 3 is the key. Okay, so now we really want to compute the cluster centroid. How we did that is we can do a command called group by. You group by this last column you just added in. Group by means, and then after grouping by, you need to do an operation called mean. Mean means just take the average. Okay, so what does this group by and mean actually does? Group by means you subdivide the big data matrix into pieces by the column you assign, right? So in our case, cells belong to 3 clusters. So you subdivide this big data matrix into these 3 sub-matrices. And then after subdividing, you apply an operation called mean. Mean just means squash this each of these submatrices into a shape like this. Right? When we think about matrix operation, it's always good to think about the shape, how a chunk of data becomes a different shape. So mean means you take the mean or the average of all the cell belongs to this submatrix, subtable, or whatever, and calculate the average average for every gene. So for this gene, this reduces down to a number. For this reduces down to a number. So this entire subtable will reduce down to a row. That's the cluster profile for every gene. Okay, you do the same for all the sub matrix, and each one will turn into a row. In the end, you combine all these three together. So that should give you 3 rows. Okay. Okay. Merge them all together. Okay. So this group by the mean step, one step does this whole process. Okay. You were given this big matrix, you subdivide it into parts. For each part, you do something to summarize it, then you merge them back together. So that's group by the mean. Let's see if that makes sense. So this temperature grouped by mean. Now let's call the new thing centroid, because that's really the centroid we wanted to have. Okay, now this is the centroid. It has 3 rows, 483 columns. Does that make sense? It's exactly what we want it to be, right? For each cluster, the level of expression. Do you see any difference here? Just by scanning through the numbers, you no longer see zeros. Initially, you see a lot of zeros. Now you don't see any zeros. That is just because initially a lot of cells don't get transcripts being detected inside. But if you group lots and lots of cells together, any gene will have some expression on average, right? So that's a way of reducing noise or combating what was it, dropout rate, in capture efficiency, and all of that. So then we wanted to visualize this centroid. That's the step 5. We wanted to z-score the results and show it in a heat map. So why do we want to z-score the results? Let me just don't do the z-score and show you what it looks like. Okay, in order to show this result, we just need to plot a heat map. Or a fancier way is a cluster map just to show the structure. So I just do a cluster map of centroids. It's too big to show here. Maybe I can try change the figure size. Sorry, the other way. Okay, so here's my centroid. 3 rows, a lot of columns, right? Does it make sense? I mean, it makes sense in terms of shape, but you see a lot of dark colors. Why is it a lot of dark colors and a few non-dark color? It's because all these genes in the middle are relatively expressed at a fewer level than other genes, right? Usually when you show a heat map like this, you don't want to compare between different genes or the absolute level of expression for different genes, because that's usually meaningless. Each gene has its different function, have different baseline level of concentration or expression in a given cells. There's no point you can show the variation of two different genes together. So that's why we z-score things. By z-score, we mean we compare every gene relative to its baseline rather than the absolute value. Okay, so I wanted to apply z-score on the centroid. So you can just do z-score centroids for every gene. So Z cent. Okay, for Z-score, you probably need a package. So let me call SciPy. Yeah, and then that does the Z-score. Okay, so for Z-score, I imported a package and that does the Z-score. Or you could, Z-score is also easy if you do subtracted by mean divided by standard deviation. So many ways you can do that. Or this whole operation alone can be Z-score but after you do the Z-score, you just wanted to check you don't do Z-score the wrong way, right? So there are two ways you can do Z-score. You either Z-score along the column or you Z-score along the rows. What along the column means for every gene, you make their mean expression level 0. If you do it along the rows, it means for every cell you make the average expression across different genes 0. Which way do you want to do? You want to do it along the column, meaning for every gene, you want there internally, the average is centered at 0. So that's something we need to try and check the results. So this is my Z-score, and you just need to check visually that for every column is every gene, their average is about 0. That seems to be right. Okay, and then then we can do then we can plot the results. Okay, I don't know what I did to that. Huh. Okay. Let's now redo the cluster map. Now you see it makes a big difference, right? Now what it shows is you see 3 clusters, each has a set of gene being specifically expressed in that cluster. A group of genes for 0, for 1, for 2. Okay, so because I only showed 3, it's obviously under splitting. If you show it for 20 cluster, it'll show a more refined structure like this. Okay. Okay, so that's that's this exercise. Okay. Questions? So there's no class evaluation form. So I guess that's pretty much what we will do for the class. So let me just spend 5 minutes to wrap it up, perhaps to show you what are the things we can do with spatial data. So one example is you can read this paper, Fan et al. 2022 Science, in their figure 4. It's a paper about spatial transcriptomes, where they showed spatial neighborhood enrichment analysis. They ask a simple question: are some cell types, once you get cell types, you know their spatial location, are some of them closer to each other than others, or are some of them distributed randomly in space? Can we get at this using the data set we have? For example, we have this data, 20 clusters, distributed spatially in the brain section. We ask, are any pairs of those 20 clusters tend to be close together in space? Or are there any pairs that tend to avoid each other? Are there types of cells tend to avoid each other for some reason? Or are the same type of cell try to be colocalized, or are the same type of cell try to spread around across the space, each have its own territory, right? You could have imagined vascular cells. In a sense, they need to be close together to form a wall or to form a pathway, but also they need to spread around quite uniformly in order to support brain, right? But how do we get these spatial enrichment questions? So, and how do we compare it with a baseline, right? So there is a simple trick, but often quite helpful and powerful, is you wanted to shuffle your data in order to get a baseline. By shuffle means, what if we randomly assign each cell a cluster in space? So rather than each cell having a decided cluster generated by k-means, we randomly shuffle the cell location. Again, the cell labels, they assign one among the 20, such that each cell in space you see randomly get a labeled color. After shuffling, so this shuffling preserves the spatial location of each cell to begin with. It preserves the number of clusters in total, the number of cells belong to each cluster in general, but it destroys which cluster belongs to which cell, right? And then that generates a baseline. So then if you compare for each pair of clusters, so 1 out of 20, 1 out of 20, for each pair of clusters, how many times do they tend to be neighboring each other? This is your raw data, and compare the raw data with the shuffle data. In your shuffle data set, how many times do each pair of cluster tend to be neighboring each other? Then comparing between the two, you know whether the actual data rise above the chance or below the chance, whether certain pair of cell types are more likely to be each other than random chance or tend to avoid each other, right? So this, essentially, if you calculate the observed cluster pairs in space versus the expected, meaning randomized pairs in space, you can calculate, divide the two, you will get the enrichment ratio. Whether for any of these pairs, are certain pairs more closer together or depleted with each other. So that's this enrichment ratio thing you can play with. So I provided you code with that to play with, and that's one thing. Another thing is there are many tutorials online. I guess I serve the Squid Pie is one of them, and it has some standardized pipeline to do probably any of the steps we talked about in class, and more fancy ways of doing things. And they have some tutorial data for you to play with. Just check their tutorial, analyzing different data, seq-fish data, slice-seq data, whatever, okay, merfish data. Well, so they provided a tutorial usually in the format of the Jupyter Notebook, the same format we used in class. You can just download their code and run it yourself and play with it. Okay, that's how you can learn. Yeah, that's it. That's the end of the class. If you have more questions, I'll stay here for a while. Otherwise, good luck for everything. Okay. I'll send out the homework, and it should take less than an hour to finish everything. It should be easier than all these assignments. Okay, thanks.
Thank you.
Thank you.
So we just have the homework, and then was there a quiz or just the homework?
Yeah, there are two pieces, a quiz
Okay.
and homework. Yeah.
And you'll send both.
Yeah, I'll send both. Yeah.
Okay. And
That would be a
that'll
link.
be sent like today.
Sorry, can you say it again?
You'll send that today.
Yeah, I'll send that today.
Okay.
Yeah.
And then it's due, you said what day?
It dues next Friday, but
Next Friday. Okay.
yeah,
Okay.
but we are quite lenient until Eloy asked me. So
Okay.
yeah,
Thank
but
you
try
so
to do
much.
it before next Friday. I think if you
Okay.
can squeeze 1 hour out of it, you can do. I'm sure you.
Okay.
Yeah, okay.
All right. Thank you. Bye.
This was really helpful.
Thanks. Okay.
Oh, it's not over.